Well, it's a pleasure talking with you today. Laura asked me to talk about this topic, gene therapy. Sorry. Give me 1 second here. That was the wrong talk that was up. Now we've got the right talk. I was working on multiple talks. To talk about gene therapy for skeletal diseases. And, this is a different sort of stage of talk compared to, I think, what is normally talked about in this series. And part of it is because we are not yet truly clinical yet. But hopefully you all appreciate that we are on the verge of clinical. I'm going to touch on this a little bit. And also I'll touch on an example at the very end of our own work, which is in clinical trial, and how it could impact, in fact, rare diseases. I want to give you just a little bit of perspective. I started my training as a postdoctoral fellow with Arcodet at Baylor in the mid-1990s. And at that time, we really felt gene therapy, it was in the midst of the first sort of phase of the hype of gene therapy, was going to cure every single disease. And the investment in the field, I think, reflected that hype. There was reality in that, but obviously there was a lot of hype. I initially started focusing on inborn errors of metabolism and liver-directed gene therapy, and lived through the period when that hype crashed, or the hype was burst, so to speak, or eliminated, when it became clear that there was no panacea, and that the Jesse Gelsinger episode with the death of Jesse Gelsinger from a systemic gene therapy trial sort of totally almost destroyed the field and went the other way. Again, it really underscores how the reality beyond the hype lies somewhere in the middle. And I was also on the FDA advisory committee circa 2017, which recommended the approval. It was a 16-0 vote of the first ever in vivo gene therapy in humans. It was a Spark Therapeutic AAVI gene therapy for a very rare inherited form of retinitis pigmentosa. I remember sitting in that room saying, my goodness, it's been over 30 years or circa around that period of time. And, and it's becoming a reality. And I think that we are now seeing a flood, hopefully not with the same degree of hype, because there's actually science and data and some reality base in terms of the investment, both financial and scientific, into what is possible. But we are now here finally, which is amazing. And I remember giving talks in the late 90s and early 2000s, and the question is, well, when will it be in clinic? And I could never answer that question. But when you think about how long, quote, drug development takes, it's probably not unreasonable, and especially of a class of drugs which was completely new and we needed to learn a lot about. My own personal journey during that time was we continue to work on gene therapy, albeit not for liver diseases, and really in the past decade have applied it to more skeletal diseases. And I think that what we will see, and I'm happy to discuss this, I'd be interested in each of your personal views about where would be the greatest application, because, as you will see, there are challenges with targeting the skeleton, unlike tissues like the liver or the back of the eye or the muscle. As was mentioned by Laura, I'm happy. I view this as an interactive, since we're not going to have a case discussion. Please, if you want to make a comment, either just interrupt me. I would be more than happy to pause to discuss and clarify some points, or just give you my opinion on certain points that may be relevant. You can either put it in the chat, and Michael promised that he would monitor it and interrupt me if someone had a comment or a question, or again, just interrupt. It's completely informal. We're all among colleagues and friends here. Okay, so let me get going in terms of my topic. Now, here are my disclosures. Relevant to this talk, we have licensed our gene therapy for the treatment of osteoarthritis to Pacira Biosciences and Genequine Therapeutics, and they are performing clinical trials on this, and I will show some data from them. That's probably the most relevant disclosure in this context. So when you think about therapeutic modalities, just generally speaking, whether it's common or rare diseases, what we're most familiar with is classes of small molecules, all sorts of small molecules. You go bigger, and of course we have proteins. And certainly in genetic diseases, the first successes came in the context of enzyme replacement. I think we all remember when the first enzyme replacements were developed, much from the work of key individuals at NIH in the 70s, and but then implemented in the period of the 90s, especially, and the late 80s, really proved that you could deliver and replace a missing protein in the body effectively in the context of a rare genetic disease. Obviously, all the rage, in terms of, instead of replacing, inhibiting antibodies as a therapeutic approach. And then, of course, for the past, again, 30 plus years, the idea of clinical gene therapy. And this has been the idea of delivering genetic information, whether it is DNA and now all the vogue RNA, required some type of platform. And that is in the context of viral, mediated by some modified virus, or it could be non-viral, oftentimes encapsulated in some type of carrier. And of course, the applications of direct gene editing, which again is not new. Folks have been trying to do this. We even try to play in this game decades ago, but never had success because of specificity. Of course, that has changed all now with CRISPR-Cas9 because of specificity and or efficiency. And at the end of the day, gene therapy is no different than any other therapy. It's about how efficacious and effective it is and how toxic it is. And also relevant to gene therapy is stem cell therapy. Now, obviously, we can give stem cell therapy without genetic manipulation, but there is potential for specifically autologous stem cell therapy with gene therapy and or genetic manipulation to, for example, avoid some of the major issues about the host immune response and the donor immune response. The goal, of course, is replace, put back what's missing, eliminate, get rid of something bad, or ultimately with gene editing, correct the underlying mutation. And so how is most gene therapy done? And so this is sort of, again, there are many ways one could imagine it, but these are the most common. Adeno-associated virus is a very popular virus platform. It, as I alluded to, was the platform for the first ever in vivo clinical gene therapy approved by the FDA for the genetic form of RP. Since then, other approvals have come, for example, for spinal muscular atrophy. Adenovirus, widely used, a platform we still use, but primarily in the context of cancer. And certainly many of you on this call may have gotten gene therapy with adenovirus in terms of immunotherapy, if you had the J&J adenovirus vaccine. And then, of course, oncoretrovirus is integrating viruses, which are especially useful in diseases or that target tissues which replicate quickly, like the hematopoietic system. And the best example of that is the treatment of severe combined immunodeficiency and the studies around that. Now, many applications for non-viral, be it DNA or RNA, but again, often requiring some modification and or carrier. Modification and carrier to target it to a specific cell and tissue to have efficient uptake, but also importantly, to potentially escape the lysosome. Because again, the normal process of taking up material from outside of the cell often is to direct it to the lysosomal degradation pathway. And there are many molecular tricks that can be done, either in the carrier and or in the modification of the nucleic acid to help it escape the lysosome, which is again very important if you're not trying to treat a disease like in the lysosome. I mean, one of the reasons why, for example, enzyme replacement therapy was the first real example of that working, of protein therapy being effective, is because it works in the natural place where the cell will take up that enzyme, which is the lysosome. That's not surprising. So gene therapy, again, is probably no different than any other type of therapy or real estate. It's location, location, location. What are we trying to do? Are we trying to target whole organs? In the case of skeleton, obviously, it's especially challenging because we're targeting a very large organ. It's not one isolated place, such as the retina of the eye. It's distributed and highly vascularized. Multiple tissues within that organ. The organ of the skeleton, broadly, described, as we all know on this call, it contains many different tissues and cell types. So do we go about it with local versus systemic therapy? That's an important first question. Well, it depends on the disease you're trying to target. If you're trying to fix fracture healing, well, local may be sufficient. If you're trying to fix osteogenesis imperfecta, well, you need something that hits most of the bones, and as well as other tissues potentially. If you're trying to treat, osteoarthritis in the knee, well, there you can have localized intra-articular systemic injection. So important what you're trying to do. Where, mostly it's either in the body or outside of the body, right? So in vivo or ex vivo. The first approved gene therapies was actually ex vivo. The AAV-RP treatment was an in vivo gene therapy, but for many years, there have been studies leading to approval of things like CAR T cells, right? Where you take the cells outside of the body, manipulate them ex vivo, and reinfuse them. This is important in thinking about disease process. This is why all the basic translational research leading up to thinking about therapy is important. What is the cell autonomy of the disease process you're treating? Are you treating a protein that sits within the cell in a compartment of the cell versus a protein that is secreted? The pharmacodynamics and the pharmacokinetics are dramatically different and unique to every disease. And the only way to really be predictive in the preclinical space is to be able to think about the cell autonomy and what that tells us about correction. And as I touched on, how much is related to that question? A intracellular protein versus a secreted extracellular protein will demand different levels of correction. I remember giving a talk, even as a trainee in 1995 at an FDA gene therapy meeting. And, I was trying to address this issue of liver. Do you need to hit all the hepatocytes, 5% of the hepatocytes, 15%? At that time, we were targeting urea cycle disorders. And so every disorder had different requirements. And treating a disorder like hemophilia by targeting the liver is very different than treating a lipoprotein disorder by targeting the liver in terms of how much protein you have to produce and how many cells you have to infect or transduce if you're not using a virus. When is important? Is regulated expression important? Can you just pump out a ton of what you need? When you think about transcription factors, for example, we know, and there are now many genetic experiments, when you look at transcription factors of the skeleton, to pick two, SOX9 and RUNX2. We know that loss of function causes disease. Well, it turns out that if you overexpress SOX9 and overexpress RUNX2, you also have abnormal cellular phenotypes. It's not surprising. Proteins which have to be exquisitely regulated, like transcription factor, have key, context, developmental, and physiological functions. So, it's not as simple. I want to make cartilage. Let me just pump out lots of SOX9. It will have a negative effect, and some very elegant genetic experiments using transgenic animals have been done over the past 3 decades that really support that. Finally, toxicity, host immune response. That's the bugaboo of every therapy, right? What is the toxicity of the therapy? And especially with gene therapies, you add the potential for genotoxicity if you're actually integrating in the genome, which is the case with some of the oncoretroviruses, less so with viruses like AAV and adeno. But in the case of gene editing, we are inducing a genomic targeted event, and off-target events can be important. Now, what I would say is, in the context of safety, I think there have been lots of experiments done going back to the retroviral gene therapies for severe combined immunodeficiencies, that one of the things which is important in determining toxicity is whether the genotoxic event occurs in an area that provides a cellular proliferation or survival advantage. And I think when we heard about the early SCID trials where there were development of cancer, part of the reason was not because it integrated into the genome, but it integrated into the genome in a place which provided a cellular survival advantage in the context where the uncorrected cells had a survival disadvantage. And so you basically are doing a genetic screen for cancer. So I think that's important in thinking about not just whether there is genotoxicity, but is there a predilection for places where you could select for a cell type because of the nature of the underlying genetic mutation in other cell types. And we certainly could talk more about that. So really, to be able to ask, is a disease a good target for gene therapy? These are the things that really have to be considered from a clinical, preclinical translational perspective. Any comments or questions? I'll pause there. This is, for me, a very important slide. And in decades of doing this and thinking about it, this slide has never changed. Okay, so what are the players? What do we have in terms of gene therapy delivery platforms? On the non-viral space, so no viruses, liposomes, and lipid nanoparticles, as now they seem to be very hiply called, are all the vogue. Many of you have gotten a dose with the COVID vaccine of some lipid nanoparticle encasing messenger RNA. Problems are efficiency and specificity. So for a vaccine, it's not a problem because you can give a tiny little dose and your immune system will do its job. But if you need to transduce 100% of the liver or 100% of skeletal cells, you can imagine that this approach may not be sufficiently efficacious or give you sufficient targeting in that context. But in general, we know that it's well tolerated. It's not super toxic. I mean, it's not that it's without toxicity. This is important. Any of these, if you give enough of, you will get significant toxicity. Toxicity is almost always dose responsive, right? And so that's important to keep in mind. You can give repeated administrations because the platform itself doesn't stimulate an immune response that clears it for future. So it's not as if you can get a B-cell mediated antibody response to the lipid. And so that will prevent its re-administration, so that there are advantages there. DNA as sort of the genetic information is actually efficiency. And it's interesting. Decades of work has been going on back to the 90s of trying to use DNA for delivery. It's very simple. The idea of giving DNA and then it would be transcribed and you make a protein, it works, but it's not very efficient. I mean, any of you who've done experiments in the lab know when you do a transfection of a plasmid into cells, it's actually highly inefficient. And you actually only get a signal from a very small amount of the actual DNA you would deliver. Now, RNA, on the other hand, is very efficient. And, it's not surprising. You're bypassing the transcriptional requirement there. And, your cells are designed to translate protein from RNA. So this is why RNA, and again, not new, has been always thought of as a very effective way of doing things. But why not? Why did people jump on this 10, 20, 30 years ago? Well, because pre-COVID, you could never imagine having enough minus 80 freezers in every Walgreens or CVS to make true clinical translation a reality. And that's also important to keep in mind. If we're thinking about gene therapy for 100 people in the world, then it doesn't really matter how expensive or how complicated it is. You're trying to treat a disease with 1 billion people. Things like logistics can really prevent it from happening. One of the advantages of viral gene therapy has been evolution. So shown on the right has taken advantage of developing what are basically missiles for delivering nucleic acids. That's what adenovirus, adeno-associated viruses, retroviruses, lentiviruses, herpes viruses were evolved to do. So one could argue, while efficiency has always been an issue on the left, it is not an issue on the right. Now, that's a generalizable statement. It depends on, again, the cells that you're targeting. But I don't need to tell this audience that an adenovirus very effectively, depending upon serotype, will infect every pulmonary bronchial epithelial or nasal epithelial cell in your respiratory tract. And that similarly, herpes virus is very efficient in infecting neuronal cells. So one of the advantages of viral vectors is that they have been under literally millions of years evolved to do one thing: infect the cell. Now, the challenges, of course, on this front is that we have also evolved millions of years, well, maybe not millions of years, but many years, to develop the immune response against these viruses. And that's where the challenges of toxicity become a problem. When you think about viral vectors, there are many applications which are either approved or on the verge of approval or in deep study clinically. I touched on adenovirus in the context especially of cancer. And this is especially true because adenoviruses are very efficient. And in cancer, you want to infect every cell possible. And they can be quite immunogenically, systemically. And in the context of cancer, the immune response is a plus, not a minus. Adeno-associated vectors have been evolved and developed over the years to target especially cells which are non-replicating and quiescent, like brain cells and retinal cells, and more or less liver cells. And again, RPE65 was the first example of an FDA-approved therapy. Same for SMA. Retroviruses, especially useful because they integrate into hematopoietic cells. They have been used for development of CAR T cells, for example. And lentiviruses, in the family of oncoretroviruses, can also infect non-dividing cells as well as dividing cells and have been used in contexts like the brain as well as hematopoietic systems. Herpes, because of its neurotrophic nature, has been especially a target for brain-related applications and cancers related to the brain especially. When we think about viral platforms, this is sort of a nice comparative picture, schematic of adeno-associated viruses, a very small virus, adenovirus, a capsid, nucleocapsid and capsid-based virus, and then lipid envelope retroviruses. And you can sort of get a comparative size. Now, what is also important to know is the size of the platform corresponds to the transgene it can carry. So AAV can carry up to maybe 4.7 kb of cargo. In a specific type of AAV called self-complementary AAV, which can express the gene much more quickly than traditional AAV, you even have less space, about 2.5 kilobase. So it's really tiny when you think about it. Adenovirus, because it has a much bigger genome, can carry up to 36 kb. And the version that we have evolved here at Baylor for many years and we continue to work on, something called high capacity or helper-dependent adenovirus, can in fact replace the whole genome with your transgene. So that's a lot of space. It's also useful for not only big genes. So for example, let's say you want to treat Stargardt's disease, which is ABCA4. Well, that's a 12 kilobase, or bigger cDNA. You're not going to fit that in AAV. So you need vectors that can hold more DNA. The other advantage of helper-dependent is you can do combinatorial therapy, which again, if you're studying or treating a more common complex disease, we know that really the only effective way to do that would be combinatorial therapy. You're treating even pharmacologically, hypertension, HIV infection. Monotherapy doesn't work usually effectively. So that's important to keep in mind. And retroviruses have, oncoretroviruses have a space of about 7 to 12 kb. Something to just, again, think about in terms of delivery. And that's, remember, the transgene has to include not just your gene of interest, but also the regulatory regions. And so a lot of engineering has occurred in lots of labs to try to shrink things and cut out things that you don't need to try to still deliver a bioactive product. Now, when you think about non-viral, and I'm going to focus on RNA therapeutics because I think this is where, obviously has gotten a lot of press, but there are many types of RNA-based therapeutics where you're targeting or expressing an RNA. Now, one which I personally am very high on is targeting RNA using allele-specific oligonucleotides. And again, this is not a new idea. It has been around for decades. And this is where the support of the NIH and funding has really empowered this very exciting period where I think in the next 5 to 10 years, we will see, I think, hundreds of applications. ASOs will bind RNA and can either lead to its degradation or inhibit its translation. And so there are clinical trials, there are drugs that are FDA approved using this approach. So as a class, we now have a lot of experience with them. And I think it's going to be growing more and more. We have a collaboration with Ionis Pharmaceuticals in this space. And this is, again, I think, a real exciting domain for different therapies. Sort of RNAi or RNA interference, where the moiety is not a short DNA molecule, but actually an RNA. So a short interfering RNA or a microRNA can be used to also similarly promote the degradation and or inhibit the translation of a target messenger RNA. All of these still would need a way to deliver this nucleic acid into the cell type of interest. Now, the direct messenger RNA, where you're actually expressing a protein or antigen, of course, this is our COVID vaccine. This is a nice example where you use messenger RNA directly, either on its own or perhaps even delivered to a cell, and you then deliver the cell to pump out the protein of interest. And again, I think there are many applications of this depending upon what you wish to target. I would just touch on stem cell therapy because it is sort of often grouped in terms of gene therapy. Now, obviously, you do not need to genetically alter the stem cell. As many of you know, this has been around since the 60s. That's what a bone marrow transplantation is, but they're now purer stem cells that can be purified. And of course, there are stem cells from many sources, and there have been lots of studies. The issue always with stem cell therapy is identification, purification, and importantly, engraftment within a larger organ structure. And I would say, you know, when we think about stem cell therapy for the skeleton, this I see is still a major obstacle. One of the most important things that was known, even from the 60s of bone marrow transplantation, to move someone into a new neighborhood, you have to get rid of everyone who's living there. And this is why very, very significant preparative regimens are done to kill the resident bone marrow. When we deliver a stem cell into the skeleton, unless that niche, that space is made, there will always be issues regarding to efficiency of engraftment of that stem cell. Now, there may be tricks one could do, and one can combine it with gene therapy. And whether it's an RNA base or a viral base or a DNA base, you know, one could do that. Now, where that is often effective is a disease where the target niche already has a survival disadvantage. A beautiful example being severe combined immunodeficiency. There are the class mutations that impact and cause skin lead to loss of those cells. And so there you don't actually have to eliminate the resident cells because they will have a survival disadvantage. And therefore, as long as you deliver a cell that's, quote, corrected, either by gene addition or gene correction, they will eventually overtake the population with the, quote, wild type cell. So again, important things to consider in any context of gene therapy. This slide is sort of an old slide, but you can see the growth of gene therapy trials. And going from, you know, when I started my postdoctoral training in the mid-90s to, you know, what's happening in the world in the late 20 teens. The number of in vivo gene therapy trials, so you can see much lower than the number of total gene therapy trials. And this just reflects that from a safety perspective, ex vivo trials are often have a hand, you know, sort of an advantage. And so especially with the hematopoietic system, but you can see the growth of in vivo gene therapy trials where we're delivering the material directly into the body. There is an enormous growth, and you can see the sort of the hype component growing again of industry and investment versus sort of academia, which has been stable over the years, and joint partnerships. So this is where there's enormous investment going on. In the recent American Society for Cell and Gene Therapy, I can tell you that at the valley of gene therapy in the early 2000s, after the Jesse Gelsinger incident, there may be 1,000 to 1,500 people attending the meeting. In the recent meeting in LA, there was over 8,000 people. I mean, it just again tells you sort of now where we are again in growth and investment. The vast majority of trials are in America, as well as in Europe, but also in Asia. It's not surprising in terms of the distribution of gene therapy trials. And the indications. So the vast majority still are in cancer space. And that's not surprising because of the risk benefit. But monogenic diseases, many of which we're interested in here, are increasing. They're about 12%. Cardiovascular, infectious, and are another tranche, and then followed by some of the other more niche applications. The types of vectors. So lots of people have been infected and treated with these vectors, and the level of safety and experience has ramped up enormously. This is why, you know, there was not a lot of safety concern when we were proposing to inject, you know, literally hundreds of millions of people with recombinant adenovirus. because there was a lot of experience over the years with gene therapy trials. The vast majority are still adeno and retroviruses, plasmid DNA from especially early on, lentiviruses, adeno-associated viruses, and so forth, in terms of some of the platforms that have been used in clinical gene therapy trials. The different phases of trials. So, as I said, there are now multiple approved drugs in the order of, tens, if you were to count both viral and non-viral, but lots of studies in phase 1. Look at the number of phase 1 studies. 2, 3, 3 obviously is later, but still a significant number. This is circa 2021, so older data. 140 in phase 3. So this is sort of the basis in which, I make the prediction that there will be, many approvals coming up through the pipeline. And also because I think as we understand the mechanisms of disease and have more appreciation of the limitations of the vectors, it wasn't just sort of wildly pushing forward into clinical trial because of more hype-based or hope-based as opposed to scientific-based translation. There are types of proteins that are being delivered. So receptor, antigens, cytokines, deficiency meaning you're just replacing a protein, suicide types of therapy, gene therapies to kill cells, on and on. So, a pretty big distribution of different types of proteins in clinical trials. When you look at our history, I really like this because this gives us a sort of a historical context of the key milestones that have led us to where we are today, which I think is, again, such an exciting time around this area. Going back to when the idea of DNA being translated into RNA into amino acids being discovered at the NIH with folks like Marshall Nierenberg, to the idea of manipulating that DNA in terms of genetic or genetic engineering, replicating DNA as a plasmid in the 70s, to the really milestone work of Michael Blaise and French Anderson, where they did the first gene therapy clinical trial at the NIH using a viral vector, in this case, a retrovirus, to treat SCID. And I remember that when that was all over the New York Times back in 1990. And then, concern around gene therapy, circa 1999, after the death of Jesse Gelsinger. And that then, triggered, a host of, monitoring, regulatory, and sort of self introspection issues, which, again, oftentimes it become extreme and inhibited the field further. But it picked up again, especially in 2000, when the clinical trial using SCID showed really efficacy for the very first time. The original SCID trials that were done by French Anderson and Michael Blaise were pioneering, but didn't really deliver on the efficacy. Here, it was actually efficacy after gene therapy. So I think this was a real milestone in 2000. The first ever approved gene therapy product was actually in China, and it was to deliver P53 back into a cancer. I don't think it really worked, but certainly it was a milestone in the world in the sense that there was a adenovirus-based delivery of P53 to treat cancer. Another milestone was the EMA's approval of the first AAV-based gene therapy. So here now is not adeno, but AAV for lipoprotein lipase deficiency. Unfortunately, this was, as often the case, where there was so much, hope and hype that it really, and I won't get into all the specifics, but, it's one of those situations that's akin to, getting the older, not the older, but the previous version of the current excitement around the current Alzheimer treatments, where, the trials only showed perhaps a signal of efficacy after you do a post hoc stratified analysis. And it was approved based on that. And of course, ultimately it didn't pan out. So this therapy, which was the first AAV approved gene therapy, was retracted. And then I alluded to really a milestone in 2017, where the FDA approved the first ever in vivo gene therapy for RPE65 mediated RP, which now has multiple other approvals in that space. Now, at the end of the day, one of the major issues with gene therapy is toxicity. And it's the problem of the host immune response, which is the issue against all therapies, but especially viral-based gene therapies. When we think about preclinical evidence, we often can see many demonstrations of efficacy. And, sometimes that efficacy translates, and sometimes it doesn't. The main issue, though, which often kills the development, is the host immune response. And this is a review I wrote many years ago when we were tackling with toxicity. And I'm not going to go through all of this, but the bottom line is, the take-home message is, Mother Nature is really smart, and we have evolved as organisms to tackle pretty much every pathogen there is. And if we are trying to now fight that with delivering a pathogen, quote, of our own, one can imagine the potential problems. And oftentimes it has been the adaptive immune response, and that was one issue, whether it was T cell mediated or B cell mediated. But then we became aware of the innate immune response, which can be a problem and occurs in all situations. I see that there is a text. Michael, you're supposed to monitor this for me. I see Michael Collins was asking, has anyone delivered genes or ASOs to specific bone cells? So in vivo, not specifically that I'm aware of. Obviously, there may be work I'm not aware of. Obviously, in vitro, that's not an issue. But in vivo, ASOs are not specifically targetable. Now, one could try to package it with potential strategies like ligands, which would, if you were to encapsulate it in a lipid nanoparticle and put a ligand on. Yeah, this is not a new idea. It's been around and replayed over and over again in different contexts. We tried to retarget adenoviruses years ago. It works, but again, it's about how efficient. You might get 3, 4 fold 5 enrichment. You might get less. Targeting in vivo is also problematic because whenever you target a ligand, invariably there's other interactions, especially in the reticuloendothelial system, i.e. the liver and the spleen, which will love to suck up things like this, any receptor you might develop. So again, not impossible, but there are very, very few truly cell type specific receptors that you can say I'm going to target. So you got to target other things. Again, I'm not aware of a clear in vivo demonstration where one could say I've targeted skeletal stem cells versus osteoblasts versus osteoclasts. And I don't think we'll see that. I think you're going to have to live with off-cell targeting with the skeleton, and partly because of I think the nature of the skeleton and where it's embedded and the proximity of all these cell types. So I do think while you could get preferential uptake, I have not seen, and I would bet against someone easily doing that. Now, having said that, I think that would be a transformative discovery. If tomorrow you or someone else says, I've got X, and this will hit, just the osteoblast, and it can happen. If you think about bisphosphonates, that's the oldest small molecule example of specific targeting. And that's not because of obviously a ligand interaction, but that's the nature of a matrix and what cells can resolve that matrix. So I don't think it's impossible. I'm not aware of one. If someone else is, please put it in the chat. I certainly would be interested. Okay. The effect of tissue-specific expression. This is a very old paper that we published, 1999, but I think it still has an enormously important point, which is where you express your protein of interest can affect the host immune response. This is a mouse expressing human alpha-1 antitrypsin from a ubiquitous PGK promoter from the liver, and this is one expressing it from the albumin promoter. So this also gets at a very relevant question. If you accept, you will get misexpression of a protein in a cell type. If you misexpress it in the wrong cell type, you can stimulate a very robust host immune response. And the liver is especially a beautiful example of it because here you have hepatocytes sitting next to immune cells and immune presentation cells, i.e. Kupffer cells. And so again, I think, these are the complexities when you start thinking about the skeleton, for example, where will you be expressing your protein of interest? And if you express it in an immune presenting cell type, which there are lots of in the skeleton, would it have a negative host immune response? And these are some of the things that you can do all the preclinical experiments you want. You can prove it may or may not happen. You won't know until you go into patients. This is part of that SCID experiment, which was so impressive, where one could reconstitute, hematopoietic derived cells long term, and was a landmark. Now, many of these patients developed cancer, and it was because of this, the idea that these cells, which had the SCID mutation, in fact, had a survival disadvantage. And so you might say, well, that's great, because then you put in cells that can survive, and this is why they have reconstitution of their immune system. The problem is, when you have this survival advantage, and because this was random integration of the integrating vector into the genome, you actually randomly integrate it all over the place, and sometimes hit a gene which gave those cells an even greater survival advantage, i.e., proliferative advantage, and it would over time actually beat out regular cells, which means cancer. So this is why, in this context, again, disease specific, because there are forms of SCID which are functional defects and do not have a survival disadvantage to the cells, that wouldn't happen. And we know that now with other types of SCID. And so this is, again, a really, I think, illustrative example. Now, to get away from that random integration, genotoxicity, which was a problem with this application, we now, of course, have CRISPR-Cas9, which I won't go into because I'm sure you guys have heard a billion talks around this. But obviously, the key issue here is efficiency and specificity, and that determines therapeutic index. And this is why this deserves a Nobel Prize, and which really is transformative in terms of genetic engineering and treatment. So I'm going to now end the talk by talking about specific applications in the skeleton. And I can't talk about all the potential ones. So I'm just going to focus on one area we've worked on, which is to try to treat osteoarthritis. Now, osteoarthritis is a complex disease related to cartilage and cartilage degeneration. It has inflammatory components, there are biomechanical components. I'm not going to go through the pathogenesis. I would say that it's not unique to trauma, it's not unique to aging. It occurs in our rare genetic diseases, in chondrodysplasias, in OI. And so our hope is that could we develop a platform to treat OA in targeted joints? So again, not systemically, but in key joints, via localized delivery of, in our case, a helper-dependent adenoviral vector. Now, there are many ways to go about it. Other folks have infected cells to try to put the cells into the joint. We have taken more of a direct in vivo approach, primarily because in thinking about treating the common disease of OA, not the rare disease of genetic OA, you're going to need a platform that can reach millions of people. And because of that, if you interpose steps like transducing cells, it's just not going to be tractable, either logistically and or commercially, unless we have another, COVID-like investment in an infrastructure. I'm going to skip this. I think you all appreciate the complexity of OA. Now, I would note that, again, since this has been a rare disease audience traditionally, OA is something that, we learn a lot about by mutations that affect cartilage. So the classical human chondrodysplasias, not surprisingly, they present with disproportionate short stature, COL2A1 being the first human gene that we had cloned back in the late 80s, focused on spondyloepiphyseal dysplasia congenita, but many others since that lead to OA early onset, but which I think are important in thinking about more age-related or injury-related forms of OA. We and others use in the animal model, surgical models, and there are multiple ones. We have chosen a whole range. We have done cruciate ligament transfection, which is a very severe model where you cause significant destabilization of the joint. A milder model, destabilization of the medial meniscus, where you cut the medial tibial ligament, and then more moderate forms like ACLT transection, transection of the anterior cruciate ligament, much like when we get an ACL tear in sports. And we have focused in this application on the helper-dependent adenoviral vector. And the reason is when you take out all the viral genes, you have a dramatic reduction of the adaptive immune response. And in fact, it acts like AAV then, when you infect a cell, it just sits there because it doesn't stimulate an adaptive immune response. And importantly, you can place lots of genes. And again, for treating complex disease, we feel you need to target many genes, including regulation. And so adenovirus is a virus, a double-stranded DNA virus of 38 kilobase in size, and it has a capsid protein outer layer. And when we gut the insides of it, we're left with the inverted terminal repeats, a packaging signal, and whatever we want to put in that 36 kb. And just to give you an example, again, this isn't a mouse, so it's not necessarily human. A single injection of a helper-dependent adenovirus vector expressing luciferase, over a year, it will maintain the expression, compared to the early generation adenoviruses where you have lots of viral genes stimulating an adaptive immune response. And this has been now seen in many models, including non-human primates related to liver and so forth. So this is a widely reproduced finding. Now, the problem is there still is an innate immune response because there's still a bucket load of protein, as well as double-stranded DNA, that's foreign, that can lead to an innate immune response. And this is actually why in every experiment related to this, you see this initial drop of expression before it stabilizes. And that has to do with the innate immune response and how the virus is dealt with, as well as how the genomes of the viruses are dealt with. Now, one of the targets we wanted to go after was IL-1RA. And this deals with the inflammatory components of OA. It's well established that IL-1 is a major driver of the inflammation that occurs chronically in OA. And there have been many, many preclinical studies showing that however you deliver IL-1RA, which blocks IL-1, this is a natural antagonist of IL-1, you get a potential beneficial effect in those preclinical models. Now, it has not been successfully translated to humans. And part of the challenge is the trials that try to block IL-1 use protein therapy, and specifically a drug like anakinra, which is a recombinant blocker of IL-1. There are many reasons why they may have failed. One may be due to pharmacokinetics of the protein. One could be due to pharmacodynamics of the action. We think another reason is the fact that when inflammation is a difficult target, especially in diseases like OA, because regeneration requires inflammation, healing requires inflammation, and in any chronic injury model, there's sort of a cycle of regeneration and injury, and then ultimately tissue destruction. We took the approach of actually regulated expression of IL-1RA with the simplistic hypothesis that you don't want to suppress inflammation all the time, only when it reaches a level, which is unclear, where it has the harmful effects outweigh the beneficial effects. And it's, to be honest, somewhat of a black magic hypothesis. But what we did do, and this was work of Killian Blues, yeah, so again, you can see the constitutive high-level expression has not really worked as well versus in animal models. So we developed this approach where we put an NF-kappa B promoter driving IL-1RA. So it only is expressed when there's significant inflammation because of the NF-kappa B responsive promoter. Now, this is published work, but this is in a post-traumatic mouse model of OA, and we did two, again, DMM and CLT. And the take-home message is irrespective of whether you're looking at ORC scoring, measures of severity of disease like osteophytes, and importantly, cartilage structure, the treatment group, which is the green, has benefits over the other control groups, depending upon what metrics you look at and what endpoints you look at. But that's the take-home message. There seems to be efficacy. And this is not shocking because, others had shown this with IL-1RA in various other models. But again, the difference is an NF-kappa B responsive promoter. And again, you can see this platform applied to not so much IL-1RA, but another model, lubricin, where we know that human mutations in lubricin causes early onset osteoarthritis. And this was work, again, going on over 10 years ago, where Mary Ryan in the lab showed that overexpression of leucin was, again, protective. And so here, addressing a different element of the OA disease process, not the inflammatory component, but a chondroprotective component, which, again, I'm not going to go into. It's been published. Others have now published on it in terms of the mechanism. But it does work long term. And again, this is a nice example of that work using a pretty 3D reconstruction of cartilage in a mouse knee joint. And let me see if I can activate the pointer. Give me a second here. Pointer, arrow options, visible. Yeah, here it is. So you can see the treated knee, and you don't even need quantification, but you can see the articulate. This is done by phase contrast micro CT of the tibial versus the femoral cartilage in terms of the preservation. So I think, it's a more of a visual example of how it can delay cartilage loss in terms of by expressing PRG4. Again, in this case, not regulated. Here, we felt we wanted the highest level expression possible of PRG4. Okay. Whoop, what happened to my slides here? There you go. And importantly, if you believe my hypothesis that OA is a complex disease, that combinatorial therapy and multifactorial targeting is important, we did that experiment. This was Adrian Stone, who was a grad student in the lab, actually tested whether combining PRG4 and IL-1RA could be efficacious. And here, she was able to show that they could act synergistically when you combine PRG4, shown here as combo, versus PRG4 and IL-1RA individually. And here on purpose, she used lower doses, sub-therapeutic doses of the other two. And on the right is more neurobehavioral study that supported that there was a functional correlate. But again, even from the perspective of cartilage preservation, you can see in these images, these representative images of the quantification below, that combinatorial therapy targeting multiple elements of a complex disease is important, something that you really could only do using a platform like helper-dependent adenoviral vectors. So this is sort of where we're at with this example. We actually had then moved forward with a large animal model. So this is, again, when thinking about the skeleton especially and cartilage, large animal models, albeit expensive, is very important in terms of moving into clinical translation. The carpal joint of the horse is very similar to the knee joint of a human, adult human. And in fact, working with Alan Nixon, we tested an equine version of the helper-dependent NF-kappa B responsive IL-1RA vector, which again showed that in the osteochondral chip model, they go in and actually drill a hole in the cartilage and then deliver the therapy afterwards, that we had beneficial effect in terms of functionally as well as histologically. And this is a really dramatic example of one of the horses. So here you see the horse carpal joint. Okay, and so you look at that range of motion here. This was done at Cornell Vet School. And here's one of the horses treated. Now, obviously, this is a dramatic example of that. But again, I think supporting this type of approach. And really, it's based on these types of large animal and preclinical that then this work was taken by industry, specifically by Flexion and then Pacira, which bought Flexion, into clinical trial. And they just presented the data from their phase 1 clinical trial with this vector, this NF-kappa B IL-1RA at the ORSI meeting in a plenary presentation where they completed a phase 1 study in 70 individuals. And here they're looking at a classic readout of WOMAC scores in terms of in these patients as an index of pain. And what they saw was, again, very encouraging. I think this is a phase one study, and the important thing was that it was well tolerated. What was interesting is that a low dose steroid pretreatment actually dramatically impacted the vector gene therapy. And so they're moving forward with this combinatorial, very low dose pretreatment with steroids, followed by the gene therapy. And I bring that up because, again, I talked about the host immune response. Almost all of the AAV gene therapies being done these days, especially in the liver, are co-dosed with steroids. And again, it's just, again, not this new idea that if you think about evolution and gene therapy, there is going to be some innate immune response, and that by using steroids to quell that, one could improve the therapeutic index by a host of different approaches. So, where are we today when we think about the skeleton as a target, right? I hope with this sort of broader talk, you appreciate the, challenges of biodistribution. There's a reason why many of the applications to date that have been FDA approved are localized to the back of the eye, targeting the liver, and in an example of the skeletal targeting here with intra-articular injection into the knee. And it's a real challenge, I think, if you're trying to target osteoblasts and osteoclasts broadly in the skeleton. This is why, again, I'm not aware of a successful experiment of that nature yet. That's not to say it can't be done. I think there are definitely ways that will be done and is occurring. I think the issue more is, what is the toxicity related with that, what I would say on target, but unwanted transduction of cells that we're not interested in. Again, a cell type that has rapid growth and division. If one is targeting osteoblasts, for example, they're turning over. Certainly the remodeling ones are. And so you will lose any platform that's not integrated, and so would require redelivery and retreatment, which is good for a commercial business plan, maybe not so good for a patient's quality of life, is going to be impacted by is there a cumulative toxicity over time? So that's, again, an important element to think about. The immune response to the therapeutic protein. This is something that we should not forget about. If you're treating genetic disease, are you treating a patient who is null for that protein? If the patient is null, as we used to say, CRIM negative, well, then in theory, you could mount an immune response to that protein. Now, amazingly, that's not, while this has always been a theoretical concern, and it is certainly an issue with some gene therapies, it has not been as such a major deal as folks had hoped or worried about, not hoped. And so I would say that while relevant, it has not been as common as one could have thought it would be, because there's so many null patients around on approach in terms of patients for any genetic disease. Are there pre-existing and or new antibody development to the delivery platform? Again, that's not so much of an issue for lipid nanoparticles, but is one for viral gene therapy. So for example, those patients that we delivered the helper-dependent ad for, they will develop a strong AD5, anti-AD5 immune response. Can we re-inject them? Certainly not systemically. Into the joint, maybe it's not a problem, because one could, for example, remove the synovial fluid in the joint, inject your vector, and then replace the synovial fluid. So again, this is where how and what you approach localized versus systemic will impact this issue of new development of antibody to the platform. Ultimately, this is the big question, systemic or local, and also unique for every disease we're thinking about. In the skeleton, what's the magnitude of the expression required? I can tell you that with the intra-articular OA gene therapy, IL-1RA, you don't need a lot of it. So we're not so worried about lots of it. In fact, lower doses of the vector in the dose response studies in the horses work just as well as higher doses. And it gets at this idea that when you're targeting signaling, where too much and too little, as I've often said, is bad, it may be there's a sweet spot for the dosing, which is not just more is better, as opposed to perhaps something like lubicin or PHE4, where I do think having a lot of it is beneficial. So this is a very important question to also think about when you're trying to develop gene therapies. And so with that, I'm going to end. I hope that was informative, and I'm happy to have a far wide-ranging discussion if anyone's interested.